My name is Jacob Kine, and this is the mitosis explanation video for the post-lab. So for the majority of the cell cycle, cells are entered into, are in the interphase phase of that, and that's divided up into three main little sections, G1, S, and G2. In G1 and G2, not a whole lot is happening besides that the cell is kind of building up, prepping for the mitosis portion of that, so it's gathering additional resources, just more mass, everything like that, so it's ready to divide. However, during the S phase, which falls between G1 and G2, the DNA within the nucleus is synthesized S and doubles up in preparation for dividing. Once the cell is ready, it'll enter into prophase. At that point, the chromatin inside the nucleus start to coil and condense, forming chromosomes. The centrioles outside in the cytoplasm of the cell will start to divide and start moving towards opposite poles of each other, and the nuclear envelope at that point will start to dissolve. Not fully yet, not quite until the next phase, which leads to the next phase, prometaphase. So in prometaphase, the nuclear envelope is all but dissolved. The chromosomes are at their most clearest. The centrioles have moved all the way to the poles and are beginning to produce spindle fibers that make their way down towards the chromosomes themselves. And at that point, the chromosomes start migrating to the center, or to the kind of midline within the cell, which leads us to metaphase. So that's when all the chromosomes start lining up across the middle of the centromeres, which is the central portion of the chromosome, begin to line up, and that's where the spindle fibers spanning from the centrioles attach. So once they're all set across that middle, the spindle fibers attach, the cell goes into anaphase, where the spindle fibers from each of the opposite poles pull hard and divide the chromosomes into daughter chromosomes, and those are starting to be pulled towards the opposite poles. Once they start making a good distance across, the cell will start entering into the telo or telophase, depending on who's saying, and a cleavage furrow will start to appear in the middle of the two cells. Not quite fully divided yet, not until cytokinesis, but yeah, those chromosomes are the daughter chromosomes are being pulled apart. At telophase, you start seeing a little bit of the nuclear envelope start to reemerge. Those chromosomes, they're kind of entering into that, almost ready to become two full daughter cells. And then near the end of telophase, it starts entering in animal cells at least, into cytokinesis, where the cleavage furrow will fully split down the middle, kind of like two bubbles. You kind of split that apart, and it reseals the membrane there, which is different from plant cells, where once plant cells have entered into that telophase portion, instead of the cleavage furrow, they'll start creating a cell plate down the middle, because plant cells have those more sturdy cell walls. And once that plate is fully developed, it'll integrate into the two separate cell walls, fully dividing the cells and creating those two separate but genetically identical daughter cells. One might ask, what is the main distinction between mitosis and cytokinesis? Why are they separate? Mitosis specifically is the replication and division of the DNA, the whole nuclear area of the cell, and cytokinesis divides the entire physical cell. That's why it caps off that. So again, this has been Jacob Kiner for this video. Thank you.